think we'll go ahead and get started. Thank you, Dr. Fisher, and everybody for being with us today. I'd like to introduce Dr. Fisher before he starts his presentation. Dr. Al Fisher serves as Division Chief in the Division of Geriatrics, Gerontology, and Palliative Medicine in the Department of Internal Medicine at UNMC. He attended medical school at Cornell as part of the tri-institutional MD-PhD program. After graduation, he moved to San Francisco for internal medicine and then geriatrics training at UCSF. He worked with Dr. Gordon Lithgow at the Buck Institute to gain training in biology of aging research. His first faculty position was at the University of Pittsburgh. In 2013, he moved to the University of Texas Health Science Center in San Antonio, where he was Associate Director for Research for the San Antonio Geriatric Research, Educational and Clinical Center, and also served as a co-core leader for their Nathan, Schock, and Pepper Center program projects. His research has largely focused on the interactions between metabolism, natural products, and the aging process. Please welcome Dr. Fisher.
All right, awesome. Thanks so much for that introduction, and thank you for the invitation to speak, and thank you to everybody that took time out from your busy day to hear my talk today. And let's go ahead and get this started. The goal of our talk today is just to kind of give you a flavor of how biology interfaces with the aging process. I think this is a topic that a lot of things that we study on all of our campuses interact with aging, and we often kind of think about that as kind of immutable, and biology tells us that that may not necessarily always be the case. So let's kind of start off with what is aging. And so thinking about it from a biologic point of view, we think about it as a natural, non-pathologic process that affects all organisms, and it leads to 4 characteristic features. So one is their characteristic phenotypic features, meaning that if you look at the picture of the gentleman off to the right there, all of us would probably, if we did a poll and said, how old is this individual? Probably very few of us would see that as a child. Very few people would vote for this individual being a teen. It's similarly to a young adult, or even kind of somebody who's in their 30s or 40s. Most of us would probably pick some age somewhere 60, 70, 80. And kind of what we're queuing in on is a lot of the phenotypes that we see there. We see he's got gray hair, he has wrinkling of the skin under the eyes, he has wrinkling of other skin on his face, and that kind of just tips us off. So it's interesting that we have kind of a built-in age detector where we're so good at being able to identify those that we are able to pretty accurately bin people into different age categories when we're out in our own personal lives. There's also a loss of reproductive capability. There's a decrease in physiologic reserves, meaning that's part of the reason why pro athletes, for example, their careers end is often not so much that new, better players come along. It's just that they get older and it becomes more and more difficult to be competitive in their sport. And that's also why when there's the marathon, there's kind of the elite category for younger individuals, and then there's like the senior and sometimes a super senior category, just because the seniors and the super seniors have a really hard time being competitive with the younger folks because of these declines in physiologic reserves. And there's also an increased susceptibility to disease. So if you're kind of a medical student and you're getting questioned by your attending, like, name a risk factor, for most diseases you see in internal medicine, age is actually a major risk factor, or sometimes even the strongest risk factor for the development of that disease. And so it becomes a really easy answer. It's like, what's a risk factor for heart failure? Oh, advanced age. How about cancer? Advanced age. How about dementia? Advanced age. And the reality is that's all right, which is that all of these diseases are strongly influenced by age. One of my colleagues points out that there's a calculator for estimating your likelihood of having a heart attack. And it's interesting is actually when you get to about age 60, a lot of those risk factors we think about, like hypertension, high cholesterol, family history, start to really drop out. And it's really the age that becomes the strongest risk factor at that point. And the current COVID pandemic has really hit home with the impact of the decreased physiologic reserves and the increased susceptibility to disease. Because when we think about who have been, unfortunately, that have fallen victim to COVID, it's really been older individuals, with the majority of the mortality happening in people in the age 65 and up group. And the reasons is that because of their decreased physiologic reserves, when they get sick, it causes a much greater physiologic compromise and makes it more likely they're going to need to be hospitalized in the ICU or pass away. And similarly, they're much more susceptible to acquiring the illness. And this is something that our work in nursing homes has really taught us during this pandemic. So, why is aging of interest to us? Like, why is it useful to have this talk? And as we just mentioned, age is a major risk factor for all of our adult diseases. It's also a major risk factor for disability and dependence. So when you think about people who live in nursing homes, sometimes it's folks that are younger that have had bad car accidents or serious illness, but it's often individuals who are of advanced age. Also, aging is really the only health-related condition that affects each and every person. So sometimes, you'll find medical students and be like, well, I'm not really that interested in asthma, for example. And since asthma affects kind of a minority of the population, we need people that specialize in it, but at the same time, we also need a lot of doctors or nurse practitioners that focus on other aspects. And in contrast, aging is something that really cuts across, and unless you're a pediatrician, it becomes real, or work with children a lot, it's really a major factor you're going to come across over and over again. And then finally, as I often point out to folks, aging has really been of keen interest to people. When you start thinking about what are themes that have shown up over and over again in movies, TV shows, literature, plays, there's love, there's grief. And with that, there's kind of the wanting to hold on to youth. There's the wanting to become younger again. There's lamenting being older. You think about the musical Cats has got talking about, there's some characters that talk about the great times they have when they're younger and how things are different now that they're older cats. This picture here is of a portrait of Dorian Gray, where you have a person who sells a soul to be able to stay young and avoid aging. Now, it could just be one where, if aging is kind of just a fixed thing, meaning it's like the coming of winter, where we can talk about it, we can think about it, we can make art about it, but there's nothing we can do, then this kind of moves from a scientific realm into other aspects of scholarly activity. But interestingly, it really does look like that human lifespan and aging are somewhat mutable. So this is showing survival curves from human populations of different times. And as we can see, the survival curve, meaning on the x-axis, we have how old are individuals, and on the y-axis, we have the percentage of folks that are still alive at that age. We can see in Africa, 3000 BC, that the average lifespan of individuals was probably somewhere around 10 to 12 years, which is, by our standards, is really shocking. And then we can see over time, look at how that mean lifespan, which is where it's about 50% survival, has dramatically increased to the current levels that are about 80 years of age. Also, the maximum lifespan that you can see down in the bottom right-hand corner, where the last individual in a population dies, we can see that that's increased as well. It's gone from about 65 or maybe almost 70 to up above 100 in current era. So we see both the mean and also the maximal lifespans increasing. Now, what are the factors that, is this really aging, or there are just other things that have changed over the last 30,000 years that we're chalking up to changing aging because people just live longer? Well, and part of this is that environment has a huge impact on the mean lifespan. And that's something we see here. It's very easy for us to think about what are things that are different now than 30,000 years ago. And it's things like nutrition, it's vaccinations, it's sewage, it's clean water, it's better maternal fetal health. And those things are all clearly playing a big role in this. At the same time, maximal longevity has increased, but it's really changed to a lower degree. And the reason is that this is really thought to be genetically determined. And a good example of this is these two animals on the right-hand side. There's just the usual lab mouse that lives about 800 days. And then there's the naked mole rat, which is the attractive critter right below it. Those descend from a common ancestor, and they moved underground, developed a really complicated social structure, lost the fur and all kinds of other things. But with this, they have a much longer lifespan, and they can live up to 30 years now. So even though they come from the same ancestor, there's been a marked divergence in their lifespan. And the naked mole rat has really become a powerful model to try to understand what are mechanisms that govern longevity, both at a genetic and a cellular level. But the important thing is that this tells us is that both the mean and the maximal lifespan are plastic and malleable and can be altered if we just knew how to do it. So this then again gets to how do we understand biologic mechanisms that may be underlying aging? And to do this was rather a challenge. And then this is a field that we've probably learned most about the aging process in the last 20 to 25 years of all of human history. And the reason is that first is that the animals of interest that we want to study have long lifespans. So humans, for example, it would be impossible to go through a tenure track faculty position trying to study human aging because of the fact that, in your 5 to 7 years, you have to become eligible for tenure. The person's only got 5 to 7 years older during that time. It's also really challenging to try to separate causes from effects. As we saw with that gentleman's picture, well, do all those changes in his hair and his skin, are those drivers of aging? Or is it like effects on the heart, the kidneys, and the brain, and other parts of his bodies that are really causing the effects on the skin? So it's hard for us to understand that. Additionally, aging is really global. So we have the problem of how do you compare, an aging tissue with a non-aging tissue when the whole body is aging at the same time. And then finally, when we think about diseases, one of the key things is that we have model organisms and we have interventions. So if you want to study diabetes, there are actually quite a few genetic models of diabetes that you can obtain from the Jackson labs that will make the animals more likely to have diabetes, more likely to have severe consequences of having diabetes or other things. Similarly, there are interventions like there's streptozin that you can give to a mouse and then it will develop diabetes within a few weeks afterwards. And so you can have a treated and an untreated mouse try out whatever intervention or do whatever study you want to do, and you've got a comparison. The mouse is exactly the same age that has diabetes and the other one that does have diabetes. Whereas the problem is we didn't have any animals that either showed accelerated or slowed aging or interventions that could alter aging to allow us to study biologic effects. But remarkably, despite these challenges, scientists have been able to identify both manipulations that can alter aging, such as caloric restriction, which is something that's kind of hot in the kind of fringe lay press right now. And what that is that if you feed an animal less, but not to the point where it's experiencing starvation, you can actually see pretty impressive increases in lifespan and slowing of visible aging. Additionally, there were genes identified in many species. So, for example, yeast and C. elegans, which is a nematode, Drosophila, which is a picture of these guys here, mice, and even humans, genes that are involved in aging have been identified. And that allows us to now have genetic models where we can compare animals that are really identical other than the fact that they have a mutation that alters their aging process. So just to give you an example of one of these genetic models people have identified, this is a figure from a landmark paper from Cynthia Kenyon's lab back in the early 1990s. And this paper was one that really influenced me when I was a graduate student and got me excited about going into the career that I have now. And so what she did is she screened through C. elegans mutants to find animals that had an increased lifespan. And so one of the ones she identified was this DAF2 mutation. And you can see right here, the white boxes are the wild type normal animals. There are two other mutants that have kind of a slightly shortened lifespan next to it. And then you see this one that really diverges out and is showing a markedly increased lifespan. It's kind of one where you don't really even need statistics to kind of see that there's something clearly different about the two of these. And it ends up being really over a doubling of the mean lifespan of these animals. Now, the remarkable thing is that DAF2 is known to lie in a genetic pathway with this DAF16 gene. And so she also studied the lifespan of that, showed that that was slightly short-lived relative to the normal animals. And then if she combined the DAF2 and the DAF16 mutations together, you ended up with all of that lifespan increase of the DAF2 mutant being lost, suggesting these guys lie in a similar genetic pathway, which is really kind of exciting because now it argues that there's some sort of genetic process you can tap into and alter both the mean and the maximum lifespan of those animals. So what does DAF2 do? Well, so remember that DAF16 gene lies downstream and it's required for DAF2 to do things. And so what folks did is they took the DAF16 gene, fused it to green fluorescent protein, which is a jellyfish protein that glows when you hit it with blue light, and then looked to see where that gene goes. And so in a wild type animal, when DAF2 is on, you can see that DAF16 GFP is just kind of out in the cytoplasm of really about every cell. And then if you turn off DAF2, like with the mutation that extends lifespan, DAF16 moves into the nucleus. And you can identify all the little nuclei in the worm there. And then folks have used gene expression technology to try to understand what are pathways that lie downstream of DAF16 that might be regulated. And well, if you take a DAF2 mutant, do gene expression on that relative to wild type normal animals, you find that there's an increase in expression of heat shock proteins, of antioxidant proteins, antimicrobial defenses. There's an increase in autophagy, which is a kind of damage and repair pathway. So really, it looks like a lot of things that people had thought about as maybe being involved in the aging process were key targets of this pathway. And so you have something that can in an integrated way, turn on multiple pathways that slow the aging process. Now, the question is this just something that's unique about worms and has really zero relevance to us or other animals that we're interested in? And so there was a really hot race back in the 1990s to identify what these genes are and what they do. And so it turns out that DAF2 is actually a protein that's analogous to the insulin and IGF receptors in people. And DAF16 is analogous to the FOXO3 transcription factor that's expressed in people and is involved in diabetes and cancer. And remarkably, these genes in us, for example, work together as part of the insulin signaling pathway. So it looks like, not only have we identified a gene in the lower animal that has a marked effect on lifespan, but it's actually something that we can trace back to things that mice and humans have. So could growth hormone and IGF-1 and insulin play a role in human aging? Well, so C. elegans only have just DAF2. They don't have insulin and IGF-1 or insulin-like growth factor. Humans have insulin and they also have IGF-1. And the way IGF-1 works is when children are sleeping, the pituitary gland releases growth hormone. This circulates through the body, particularly goes to the liver. There turns on the expression of insulin-like growth factor 1, and then it goes to places like fat, muscle, and bones. And particularly in children who are growing, it will go to bones and stimulate growth while they're sleeping. Now, is there any evidence that growth hormone or IGF-1 is involved in aging in vertebrates? Well, so there is. So there's the Ames and the Snell dwarf mice. These guys are shown in the top picture, and they lack growth hormone, and they have increased longevity. There's also the lone dwarf mice that lack the growth hormone receptor. These guys are also small and are long-lived. So it kind of argues, well, that axis does have some involvement in aging. And so folks then looked at IGF-1, which is kind of analogous to that DAF2 protein. And if they make a heterozygous mutant of them, these guys actually have normal body size because they still have one active copy of IGF-1, but they show an increase in lifespan. And then if we give mice extra growth hormone, like giving them shots of growth hormone, we end up with this big guy that's down here at the bottom, where you can see that it's almost probably at least a good 50% bigger than the normal mouse, but it actually shows accelerated aging. So it looks like we have kind of, again, this connection where having less of DAF2 or less of IGF-1 results in longevity, not only in C. elegans, but also in mice, for example. Now, is this at all relevant to humans? And this is a paper that I really found kind of fun, and we were talking about this when I was a postdoc. An author named Samaras looked at the lifespans of folks in Finland that were involved in athletics. And so particularly what he did is he looked at different athletes. And so he looked at the cross-country skiers, looked at the hockey players, the soccer players, and then the basketball players. And one thing is that the average height of people that play those different sports varied. So the cross-country skiers tended to be the smallest. The basketball players, not unexpectedly, tended to be the biggest. And interestingly, and remember, this is in Finland. These are all athletes. So it's one where they've got access to healthcare. They have, they're all athletic and have been well trained at some point in their life. Increase in lifespan as you became bigger. So the basketball players actually had an almost 7-year shorter lifespan than the cross-country skiers, who were the smallest group. And this is something that, it's kind of shocking when we think about it with people, but when we think about it with pets, for example, like with dogs, the largest breeds, so things like Great Danes, have a lifespan of about 7 years, whereas like the little Chihuahuas and other the toy dogs, they can live, upwards of 15, almost 20 years of age. But so it really argues that something about body size and the growth hormone axis does have an influence on lifespan in other vertebrates and importantly in people. So to further get at this, Nir Barzilai, who's at Albert Einstein, he created a centenarian cohort where he basically just placed ads looking for folks who lived to the age of 100, and then looked at their children for both the people that lived to 100 and their children to try to identify pathways that would be involved in aging. And one of the things he found was that among women in his group, that the offspring tended to be, they had slightly higher IGF-1 levels, which you would think would mean that they're going to be bigger. But instead, what he found was that their IGF-1, their height was actually a little bit smaller. And so this kind of created an interesting paradox of why do you have more IGF-1 and yet though you're kind of smaller in size. And so he then looked at, he used kind of an old school technique before the days of just, now you just sequence the gene, but back then they had to do another technique. And essentially what he was able to find was that there were some of these individuals had heterozygous mutations affecting the IGF-1 gene, which is shown here in this red box at the top there. And so he was then interested in, did these mutations impact on IGF-1 signaling? And he found that the folks that had these two mutations, that when he looked at their lymphocytes, as you can see in panel C there, they actually had lower levels of the IGF-1 receptor. When he stimulated those cells with IGF-1, the folks that were carrying that mutation showed less activation of the IGF-1 receptor, consistent with the fact they had less of the receptor on their cells. And then similarly, when he looked at downstream signaling pathways, such as the phosphorylation of the AKT kinase, he found that those folks that were carrying that mutation had activated the downstream pathway in response to IGF-1 less as well. And so the conclusion he drew from this was that at least some of his centenarians exhibited resistance to IGF-1, and as a result, they resembled those DAF2 mutants that we saw back in C. elegans. Now, so you may kind of say, well, okay, is all of aging just controlled by IGF-1? And, do we need to think about anything else? Well, and certainly data since then has really made it very clear that IGF-1 is not the beginning and end of aging processes. Instead, other work that I don't have time to talk about today has really found that over 100 genes have effects on lifespans in model organisms. And importantly, when you start to look at these genes and think about how they function and relate to them, it turns out there's a number of pathways. So in addition to that IGF-1 pathway that we talked about, there are things like mTOR signaling that we'll spend the rest of our time talking about. There's autophagy, there's sirtuins, which came from yeast, there's mitochondrial signaling, and there's cell senescence pathways. So it really gives us multiple ways that we can think about the aging process, and perhaps we may find that certain aging phenotypes are driven by one pathway and not others. Now, the other importance of having pathways that participate in the aging process is, could we find medications that might target one of these pathways? And the answer to that is, yep, actually we've been able to do that. So far there are multiple drugs that have been shown to increase lifespan in lower animals, and this ranges all the way from Drosophila and C. elegans up to mice. There's a program when I was in San Antonio that one of the sites was based at our university called the Interventions Testing Program, and essentially what they do is using specialized mice, they conduct randomized trials of medications that may alter aging in mice to figure out, do they actually alter aging or not? For a long time, it was actually kind of a negative study, and it was seen as a way of kind of debunking junk science. But then when rapamycin came along, all of a sudden the ITP really changed from something that was more of a debunking type of study to really a very exciting, hot area of science, identifying compounds that have aging modifying properties. And so out of this work, drugs that have been identified are things like resveratrol, rapamycin that we'll talk about, acarbose, that's a diabetes medicine, ACE inhibitors that are used for blood pressure, metformin that's used for diabetes, some weak pro-oxidants, and some anti-seizure medications that have all been shown to have different effects on lifespan. Now, when I bring this up, especially in the geriatrics world, people always get kind of anxious that the goal of medications that affect aging is to kind of keep people in a nursing home in a debilitated state, for as long as possible. Whereas I really argue that the goal of these types of agents is really not so much about the lifespan extension properties. It's really instead saying, are there ways that we can target? We know that aging is a strong risk factor for so many adult diseases, and the challenge we have is, how do we prevent and treat cardiovascular disease, pulmonary disease, cancer, Alzheimer's disease? A lot of times patients come in and see me and they're complaining about the medications they're on, and when you have a pharmacist look at the regimen and link it back, you can link it to all appropriate medications for each of those diseases. The challenge is that each of those diseases has gotten so sophisticated in terms of the medications we use that it rapidly spirals out of control for patients. What if we could instead have medications that target some of the underlying risk factors like aging that contribute to things like cardiovascular disease, diabetes, Alzheimer's disease, Parkinson's disease, development of geriatric syndromes like falls and loss of muscle mass? This might be something where you could actually take fewer medications and get more benefits from it. So one of these that kind of hit first was rapamycin. And so this is a macrolide molecule that's made by Streptomyces hygroscopicus, which is what's shown in this plate. And it was something that was found back in 1970 by Brazilian scientists on Easter Island. And where rapamycin came from is that the natives on Easter Island call the island Rapa Nui. And so rapamycin came out of that name. And it had kind of an interesting history prior to being studied in an aging contest. And I guess it kind of is one that shows that if your medication, that being persistent and kind of staying in the game is probably a key way to eventually make it to market. So back in the early 70s, it was initially studied by Ayers as an antifungal drug. And this was, however, during testing, it was found to suppress immunity. And as a result, Ayers decided not to pursue it as an antifungal anymore. Though NCI was then in the 70s during the war on cancer really motivated to find novel agents that could be used in chemotherapy. Rapamycin got studied in there and was found to not unexpectedly have a broad role in cancer. And mTOR plays an important role in a number of cancers, and so it's probably not surprising that rapamycin that inhibits that also plays an important role in cancer. But then in the early 80s, Wyeth buys Ayers. They decide to close their Canada branch of this and consolidate things at Wyeth's main headquarters. And as a result, rapamycin kind of dies off as a chemotherapeutic. But then in the mid 80s, Wyeth changes their CEOs. And now, of course, it's the 1980s. Organ transplants become very important. And somebody kind of realizes, oh, this was medication suppressed immunity in mice back in the 70s. Hey, maybe we should study this as an immune suppressant for the use in transplant. And as a result, it got reborn and came through and work published in Lancet in 1989 as really an important part of renal transplant and other solid organ transplants. Additionally, it ended up being, now it's involved in things like drug-eluting stents. So what does rapamycin do? It inhibits mTOR, which is the blue molecule down on the bottom of this picture. I often tease students when I use this slide for teaching in the first year of medical school that, you know, there's going to be a test on this entire figure. Make sure you look at it really carefully. But the incredible part is that mTOR is an important node point that connects up a lot of different metabolic and cell signaling pathways and links them to things like changes in protein synthesis, lipid synthesis, the autophagy, the production of lysosomes, and also energy metabolism. So it's maybe not surprising that the molecule may play a role in aging processes. So just to drill on these a little bit more detail, so mTOR plays a critical role in the production of proteins, and when it's inhibited, it changes the production of, there's a shift towards certain proteins and a shift away from others. It affects lipid synthesis, energy metabolism, and also the activation of autophagy and also lysosome biogenesis. So how did rapamycin get studied in an aging context? Well, so Matt Kaberlin in this top figure, they did a screen looking for mutants that increased yeast lifespan. And one of the genes that came out of that was TOR. Other folks have then went on to study this in Drosophila and in C. elegans, and they found increases in lifespan in these other two animals. As a result of this, rapamycin was proposed for testing in the interventions testing program. And as I mentioned earlier, rapamycin ended up being the first molecule shown to ever increase lifespan in mice. And this led to a paper in Nature back in 2009. Now, the thing that's really interesting about this work was, and I kind of know the backstory of this from when I was, from being on faculty, which was the rapamycin, the way they do things with the interventions testing programs, they put the medications in the mouse food and the mice just eat that as part of their normal diet. However, rapamycin ended up being unstable. They had difficulty getting it into the food and then actually getting it into the mice in a way that they could measure the levels. And so it took them a long time to figure out how to microencapsulate this and be able to make the mouse food. So long such that they actually had mice ready to go for the trial, and but didn't have the rapamycin ready. And so the mice actually sat for 600 days waiting for the rapamycin to be prepared. And so they ended up starting at a day 600, which is roughly about 60 years of age for people. And interestingly, you would figure if you started something at like age 60 and you're looking for a lifespan benefit, that you've really stacked the deck against yourself. You know, given that you've kind of at least half or more of your life, you haven't been exposed to something that potentially could make your life longer, and you're really starting it at the very end of life. But despite this, it actually ended up increasing lifespan about 9% for the males and about 13% for females. And then when you think about what their remaining life expectancy, it ends up being about 28 and a 38% increase in that end of life expectancy for these animals. So it's really a stunning result that despite, you know, all of the factors weighing against this being successful, that it was successful. They then went back to say, okay, what if we treat mice for their entire lifespan? And what they ended up was really about the same curves as before. If I kind of showed you this one and this one, you'd say they don't really look a whole lot different. And indeed, when you actually lay them on top of each other, the mice that start early at 9 months of age and then the late starts that started at 20 months, there actually is very little difference in the lifespan. Now, you might say, well, why is that? Why would that happen? Well, probably what that means is that the processes that are critical for rapamycin to target to increase lifespan don't happen during younger life. That essentially you can go through childhood, young adulthood, even kind of the middle of adulthood without having to target those pathways to get benefit. And instead, really the critical period for this compound to work is in the later years of life. And in a way, this is, you know, if you're thinking about for practice, for example, this might actually be really attractive in that you have the challenge of, if you have to take something for your entire lifespan, there's like the cost goes up dramatically. Doing a clinical trial becomes really difficult because now you have to start something in people who are young adults and follow them for decades before you get to the point where you're going to be seeing clinically meaningful benefits. And additionally, when it goes into practice, you have to convince people who are 18 years of age who are kind of like, why do I want to take a medicine? Or I'm going to forget to take the medicine because I'm so busy, you know, with my young adult life to do that. Whereas instead, it kind of argues, you know, we can, plus you also have the concerns of, you know, how would this have other side effects that might affect fertility? Would it cause birth defects? You know, how would it affect all these other things that are concerns in younger adults? Whereas if you had something that started in older life, you now have somebody who, you know, the coupling between taking the medication and getting benefit from it might actually be a lot closer in time. Additionally, you know, older individuals are starting to see and recognize the effects of aging, and so they may be much more motivated to take such a medication. Plus, you also take out the whole issue of reproduction and fertility because you're dealing with folks that are at a later point in life. Now, could it also have other benefits? And this is kind of where it becomes interesting. So this is how you study memory in mice. So there's a thing called the Morris water maze. And essentially what you do is you have a dish that has water in it, and you have a visual landmark like this star that's shown off to the side there. And you put the mouse in the water. The mice can swim, as you see down here in this picture, but you know they prefer to be on something solid and not having to swim. And I can totally understand that. If somebody grabbed me, dunked me in a tank of water, I would probably be hoping that there's some sort of platform that I could stand on and not have to tread water the whole time. And so what will happen is the mouse will go in there and they'll rapidly figure out there's a platform there. And as you do this multiple times, they'll actually learn where that platform is. And so this becomes a really effective way to look at learning. And because there isn't so much of a motivation factor because they really hate having to tread water to stay afloat. As a result, it's not so much about are they motivated to learn, they certainly are, and they certainly want to find that platform. So some of my colleagues in San Antonio used a transgenic version of mice that creates an Alzheimer's disease model, and they wanted to see did rapamycin have effects on learning in either wild-type animals that don't have this transgene or ones that do have the transgene. And so what they did is they started these mice at about 13 weeks of age, and they were 4 months of age, and they treated them for 13 weeks. And then they put them in the tank to do swims. They did 6 of them a day for over 4 days, and then they looked at how long does it take them to find that platform. And kind of what we find is that the first time they go in there, you know, it takes a while for them to find it because first of all, they didn't know there was a platform in there. They have to discover it and then figure out they can stand on it. But over time, we can see that they take less and less time. They remember where that platform is, and they go looking for it when they're in the tank. And what we find is that, not unexpectedly, if you have Alzheimer's disease, that your ability to figure this out is lower than if you're a normal mouse that doesn't have Alzheimer's disease. But importantly, if we treat you with rapamycin, which are either the white open circles or the white open squares, that those animals are quicker at figuring out where that platform is. Importantly, they found statistically significant differences in the Alzheimer's disease mice, but they certainly saw a trend towards an improvement that wasn't significant in the normal mice. And additionally, what they then did is that was kind of like, well, can they remember where that platform is when they can find it? The other way you can do this is to actually take the platform out and then put the mouse back into the tank and see, do they go to that spot over and over again? And so this is kind of thought to be the strongest test for memory, because basically what they're doing is they're looking for the platform in its old location. And what they found was that the mice with rapamycin, the mice that have Alzheimer's disease, were the least likely to remember where that platform was compared to normal mice. And if they treat them with rapamycin, which are the 2 bars with the arrows pointed to them, that either the normal mice that didn't have Alzheimer's disease or the mice that had Alzheimer's disease, the rapamycin treatment improved their memory and led them to go to the spot where that platform used to be much more frequently. Folks would also have the concern, like, well, if we're thinking about this for people, is it going to have problems because it's an immune suppressant? Or the other way some scientists have thought about it is, well, if this is a molecule that alters aging, might it actually get over some of the challenges we see in older individuals, such as responses to vaccines? We know that flu vaccination, for example, is much more successful in children and young adults than it is in older adults. And as a result, there have actually been the development of a special formulation of the flu vaccine that we selectively use in older folks to try to get over some of this inertia to the vaccination. So the authors of this paper did is they took old mice, and what they wanted to do is to see would it influence the production of antibody. And this was done back when H1N1 was our pandemic of the year. And so they were using an H1N1 vaccine and an H1N1 virus as the challenge. And what they see in panel F here is that if you have young mice, don't give them a vaccination. This black line that goes down to the x-axis pretty quickly, that's what happens if you take young mice, infect them with H1N1 and don't vaccinate them. They get sick and they pass away pretty quickly after getting challenged with the virus. In contrast, if you take young mice, which is this top line in panel F, you can see that if they get the immunization and are protected against it, that they don't die after being infected with H1N1. In contrast, if you have old mice, so this is the line that's kind of down below in the dotted line, that if they get immunized, it doesn't really protect them that well. And a good number of them still actually will get infected with H1N1 and die as a result of that. In contrast, if they take the old mice, treat them with rapamycin for 2 weeks, and then give them the H1N1 vaccination, we find that the vaccination works a whole lot better, and they actually are resistant now to getting the H1N1 influenza virus. And so why does this happen? Well, we can see in panel E off to the right there that the titers of antibody they produce in response to that vaccination go up markedly after receiving rapamycin prior to their vaccination. So now we've got a molecule that not only can do things like delay death due to aging, can improve memory, both in normal mice and in mice that have Alzheimer's disease, and it can also do things like reinvigorate immune cells so responses to vaccination are improved. So at this point, I'll wrap up and take any questions that folks have. And so kind of what we talked about today was, one was that aging is a universal process that impacts both human health and human function. Importantly, biology has been able to teach us that aging is not just something that we have to, that's kind of forced upon us and something that we really have no ability to alter, but instead that aging is malleable and that there are specific mechanisms that are targetable that lie downstream of this process. Importantly, being able to uncover these mechanisms has allowed us to develop medications that can either modify aging, and this could be either used as a potential therapeutic. So a lot of these things that came out of the Interventions Testing Program, we had a Pepper Center in San Antonio, and one of the important goals of that was to try to translate those into early stage human trials so that we could see how they could benefit in older individuals. Additionally, medications like rapamycin and metformin have become very hot in research as well as a way of how do you examine the influence of aging mechanisms on any biologic process you're studying. And so it's become very common now to introduce the use of rapamycin or metformin or other drugs into a research protocol to see how does aging influence very disparate things like immunology, neuroscience, gut microbiome, and other things as a way of now kind of teasing apart that aging influence. Finally, from a therapeutic potential, the benefits for people of these are currently unknown, but there are certainly a lot of avenues that exist for translation of these to the clinic in the future years, and I think this is a really promising area of research. So at this point, I'll stop and take any questions that folks might have.
If anyone has any questions, please feel free to unmute.
Hi, Dr. Fisher. My name is Kim Carlson. I'm from UNK. And I have a question for you. I found your the studies that you presented really, really interesting. I'm a fruit fly geneticist, and we have looked at aging in fruit flies. And the one thing that we see is that there's dysregulation of immune function genes as the flies get older. And this is probably in direct correlation to, when certain things happen in their life. We have a whole host of candidate immune genes that we're looking at to see how they correlate with the aging process. Do we have anything besides IGF or FOXO in humans that we also can see this dysregulation throughout the lifespan in humans?
Yeah, so meaning other pathways that, so just to clarify your question, so there are other pathways besides IGF-1 that are drivers of human aging?
Right, that are specifically related with immune function, because that dysregulation starts pretty early in flies, and I was just wondering if there's any correlation with that in humans.
Gotcha. Yeah, so that's, and you bring up a great point. So there's the concept of inflammaging. So, I think the rapamycin, that the last slide where it kind of showed that it enhanced immunity. Well, there's kind of two aspects of inflammation, which is that on one hand, the ability to make a good immune response against pathogens goes down with aging, but you also have this non-specific increase in inflammation that occurs with aging. And so in like the frailty literature, for example, which is kind of factors that happen to some older individuals, such as loss of muscle mass, loss of weight, low energy, that those individuals show evidence of elevated inflammation in the absence of an infection. In terms of like pathways that people have looked at that may be contributing to that, there's work that sirtuins may be involved in this. And then there's also evidence that NF-kappa B, which is a key driver of the immune system activation, may be inappropriately activated during aging. So specifically, like in senescent cells, one of the things that may be happening is there is the activation of NF-kappa B, the production of cytokines and other compounds that will activate the immune system. That leads to hyperactivation of NF-kappa B in elements of the immune system and in other cells. So there certainly are other pathways that touch upon the immune system.
Hi, Dr. Fisher. This is Brian North. I'm over at Creighton University. It's a very interesting talk. My lab works on molecular and cellular mechanism of aging as well, heavily in the sirtuins, sirtuin pathways. But I'm curious about if you could kind of clarify the use of rapamycin and its potential as a therapeutic in humans and the data that was generated in mice. Is it immunosuppressive in mice to the same level that it is in humans?
Yep, it is. So one study that we had, so our Pepper Center, one of the pilots was actually studying rapamycin in people. And we wanted to look, we did it for a short period of time, looking to see, did it alter like response to influenza vaccine? And actually the titers that people made to influenza vaccination really didn't alter very much between the people that were in the treatment group and in the control group. So it is an interesting, as some of my colleagues have pointed out, is that when we think about rapamycin as an immune suppressant, that's often in the context of people who've gotten a solid organ transplant. So these are folks that are often taking other medications, have this kind of foreign body inside their body all the time. And additionally, they got to the point of having a solid organ transplant because they had advanced renal disease or advanced liver disease or heart disease prior to this. And so it may be something that if you're taking rapamycin in the absence of all of these other factors, that it may not necessarily be as immune suppressant as we thought for.
Mm-hmm. Great, thank you.
Cool. Well, if there are no other questions, if you want to shoot me an email afterwards, certainly I'm happy to take questions offline as well. So thank you so much for attending today.
Thank you, Dr. Fisher. We appreciate it.
All righty, take care, everyone.
You too.
Thank you.